Hello everyone, I am Dr. Ritu Singh. Today's lecture is on Introduction of Pharmacodynamics. So we will start with the definition of pharmacodynamics. This is the study of a drug's molecular, biochemical and physiologic effects or actions. It comes from the Greek words pharmakon and dynamikos. Pharmakon represents the drug and dynamics means the action or power. Pharmacodynamics covers all the aspects relating to what a drug does to the body. So, when we take a drug in various amounts, what will be the effect of the drug comes under the pharmacodynamics. The elements of pharmacodynamics are action and effect. So, action is the how and where the effect is produced is called as action, and effect is the type of response produced by a drug. So, generally, the action is the feedback of effect. Site of drug actions are extracellular, cellular, and intracellular. Extracellular means when a drug acts outside the cell membrane. Cellular means when the drug acts on cell level, cellular level, or on the cell membrane. And intracellular refers to the when a drug does his action into the cell membrane. The types of drug actions or responses are stimulation, inhibition, replacement, irritation, or cytotoxic. So, these are generally the types of drug action. What are the responses we take from the drug? Stimulation is it refers to selective enhancement of the level of activity of specialized cells. Like adrenaline stimulates heart, pilocarpine stimulates salivary glands, and there are so many examples of stimulation. However, excessive stimulation is often followed by depression of that action. For example, high dose of picrotoxin, a central nervous system stimulant, produces convulsions followed by coma and respiratory depression. The inhibition or depression action of the drug is the phenomena of decreased drug metabolizing activity of the enzymes by several drugs and chemicals is called as enzyme inhibition. The process of inhibition may be of two types. Number one is direct inhibition, or number two is indirect inhibition. Direct inhibition means the drug directly inhibits the action of that enzyme, and indirect inhibition means when a drug indirectly inhibits the enzyme. We will discuss further detail in next slides. So here we can discuss about the stimulatory and inhibitory action of the drug. Before, as you are seeing in the slide, that before medicine there are natural chemicals, receptor sites. On receptor sites there are receptors and normal cellular activity. So when we introduce a stimulatory drug, it will fractionate or divide it into its components and mix with the natural chemicals. These stimulatory drug components binds to the receptor sites and then enhance the cellular activity of the cell. The blue dots represents the stimulatory drug and yellow dots represents the natural chemicals. These are the receptor sites where two blue dots are attached. Now when inhibitory medicine is introduced into the cell, it will divide it into its components and attached to the receptor sites and thus inhibit the activity of cell, so it will block or decrease its activity. Receptor sites, ligands, what are receptors, we will discuss in other lectures. Now the type is replacement. Use of natural metabolites, hormones, or congeners in their deficiency is called replacement. Like in Parkinsonism, we will use levodopa, and in diabetes mellitus, we will use insulin, and in anemia, we use iron. We replace the original components of body with the drugs. This is called replacement type of drug action. Next type of drug action is irritation. This connotes a non selective, often noxious effect and is particularly applied to less specialized cells like epithelium and connective tissues. Strong irritation results in inflammation, corrosion, necrosis, and morphological damage. This may result in diminution or loss of functions. Next type of drug action is cytotoxic. Selective cytotoxic action on invading parasites or cancer cells, attenuating them without significantly affecting the host cells, is utilized for cure or palliation of infections and neoplasm. For example, penicillin, chloroquine, zidovudine, cyclophosphamide, and so many more. Now, the general toxicity of cytotoxic drugs are nausea and vomiting, bone marrow depression, alopecia, gonads like oligospermia and impotency. On fetuses, the cytotoxic actions are abortions, fetal death, teratogenicity, and carcinogenicity, and hyperuricemia. For any doubt or query, please comment in the